Okay, I'll just say first, thank you so much for the invitation. It's really a great honor being here today. Seeing really the great science. Talking after Tony, it's really, really crazy over here with all the important things that he's done. But today I want to kind of emphasize a little bit of the work that we're doing in my laboratory and really get your feedback as well about how the potential of our finding going to be in tissue regeneration. So I do want to open first with one thing that I found truly fascinating is that we all have a reoccurring life event that happens each and every day, and this is tissue regeneration. We are losing and replacing billions of cells every day, and that's completely normal. The way that I like to think about it is that we all know how we woke up this morning, but we must have changed several times since then, and this is not just a fantasy story out of Alice in Wonderland. In fact, different organs in our body are regenerated in different rates. So the skin and the gut, for example, replenish themselves very, very fast. We have almost completely new intestine each and every 5 days. But other organs like the muscles replace themselves very slow, or in the adult, the other organs like the heart don't replace themselves at all. And so key players in this regenerative process, as all of you know, are stem cells, but their fate decisions needs to be balanced because too little of stem cell activity will create aging, but too much will create cancer. So essentially what we are very interested to understand is what influences stem cell fate decisions. And the skin is really an excellent model to study those type of questions because in our skin there are multiple types of stem cells, including those stem cells that creates the hair follicle, those beautiful hair follicles that you see in this picture. And it's also susceptible for systemic changes. So things that we are eating, things that happen that surrounds us really affect our skin biology. So the questions that we became interested in is how the stem cells can integrate all of those systemic changes and how they can basically affect tissue regeneration. One of the systems that potentiate that system to affect those systemic changes was the vascular system that creates a network in our body and can really affect how stem cells behave. Normally when I talk about the vascular system, and I do have the tendency to do that, most of the people think about blood vessels, and that's true. We have blood vessels that carry cells and nutrients into the tissue. But we also have another vascular system, and that's the lymphatic vascular system. That system will carry back fluids, macromolecules from our tissues back into the circulatory system and for drainage. So now we wanted to focus the question and ask how the vascular network can coordinate stem cell behavior across the tissue. One of the things that we face first is that the skin is not transparent. I can't just simply look at the vascular system as if it was transparent. So we adapted a completely new clearing technique that takes away the light refraction and enable us to look at the skin under completely non-compromised conditions. And this is the first image that we were able to see. So now this is a tissue in 3 dimensions. You can see at the base here, those are the stem cells here, and what we noticed is that there was a vessel that was very dilated. It was a big vessel that was not stained for all the blood capillaries markers. So we became really interested to understand who are these vessels. What we basically found is that the stem cells here in green, the hair follicle stem cells here in green, are tightly associated with lymphatic capillaries in red. So you can see here the stem cells, this is in 3 dimensions. We can model that type of interaction using computational algorithm. And you can see that the stem cells are really sitting on top of on the lymphatic capillaries. So now we're going to take a virtual tour into our skin to understand better about the nature of those type of interactions. So what we were able to see is here the stem cells in green and the lymphatic vessels in white. The lymphatic created protrusions that emerge from the lymphatic vessels towards the stem cells, almost as if they're holding them in place. You can see it here as well. So the stem cells, those stem cells in the hair follicle, sitting here in purple, you can't really see the marker, but those are the purple cells that are sitting there. What I've showed you so far, those are the stem cells that are at their resting state. they're actually not doing anything, they're not regenerating. Normally, those stem cells will undergo cycles of activation, so they will undergo kind of activation to stimulate hair growth, and then revert back to their quiescent state. What we do know is that those stem cells will spend half of their time growing and half of their time resting when we're young. But as we grow older and older, they will spend less and less time in the growth phase and will become more and more quiescent. And this is, in a way, a reflection of most of our stem cells in our body. And so this is something that is really interesting to understand. So while most of us making about 3,500 choices every day, stem cells needs to make much less choices. They can choose either to self-renew themselves, meaning to create more of themselves, or to differentiate and to create the tissue in which they reside in. So essentially what we were trying to understand is how the stem cells know to create the right cell type at the right place, but also at the right time when tissue regeneration actually needs to happen. So we have a completely new niche compartment, the lymphatic vessels here in white and the stem cells here in red. And we wanted to understand why lymphatics are important, so we decided to ablate the lymphatic system. What you can see clearly kind of just from morphological microscopic understanding is that the stem cells no longer small, they are very, very big follicles over here. And in the context of the hair, it's very beautiful because the mice started to grow hair precautiously at the time when tissue regeneration actually didn't need to happen. But most of us grow hair without the need to ablate our lymphatics. So what we were more interested to understand is what happened during normal tissue regeneration and tissue growth. What we notice is that when the stem cells are close with the lymphatics, the stem cells are at their resting state. But as soon as the stem cell starts to be activated, and this is based on the Ki67, meaning that the stem cells proliferate, at that time they dissociate from the lymphatics and starts to become activated. So we establish that lymphatic stem cell interaction is dynamic. It's not always, we have a completely new niche compartment, but it's dynamic. And the question that we became interested in is, can the stem cells themselves reconstruct their niche? Seeing this type of interaction and dynamic, we wanted to understand if this is something that can be controlled by the stem cells. So we profiled the secretome of the stem cells at the resting state, and that's the top kind of row over here. And what we notice is that there are factors that are important for vascular biology that are highly expressed by the stem cells, and this is specifically angiopoietin-like 7. Also using single-cell RNA sequencing, we were able to identify that this factor was specifically expressed only at the stem cells, but not in any other cell compartment within the hair follicle. To make a long story short, using genetic models and an ablation experiment, we were able to show that the stem cells themselves secrete factor angiopoietin-like 7, and that is important for the association of stem cells with their lymphatics. As soon as stem cells revert into their active state, they downregulate angiopoietin-like 7, switch program to become, to express angiopoietin-like 4, and that's reduced lymphatic drainage and ability of the stem cells to regenerate. The main take-home message is basically that the stem cells can reconstruct their niche, but I also opened this talk by telling you that different organs regenerate at different rate. And so if the hair follicles undergo cycles and work in a way of only half of their time they're active, in the intestine the picture is completely different. The stem cells are constantly active. So can it be that one niche rule them all? Can it be that the lymphatic vascular environment controls stem cell behavior across epithelial tissues? So with a very fun collaboration with in the lab of Elaine Fuchs and Dana Perry in New York, we were using, again, three-dimensional imaging of the intestine. And what you can see here in green are the lymphatic vessels, the red are the blood vessels. And what we were able to see that there is a very dense network of lymphatic vessels that emerge right where the stem cells reside in the crypt, where the intestinal stem cells are. So that really triggered our thought that maybe lymphatics are also important in the crypt, and that was true when we used stem cell marker LGR5 for the crypt stem cells. As you can see here, they were, jumped. they were also tightly associated with lymphatics there. And so no matter if the stem cells are highly activated or undergo cycles, lymphatic vessels are tightly associated with the stem cells. But maybe it's just an association, because we know that lymphatic needs to be in the intestine. They're absorbing fat and fluids and macromolecules. So maybe it's just an association. What we found is that this stem cell lymphatic connection was preserved throughout the intestine, where the small intestine or the large intestine, no matter if fat absorption was or not. And so it's not just an association of this lacteal that absorbed the fat and fluids, it's actually important for the stem cell function. What's really nice in the intestine is that we can isolate those stem cells and create organoids in culture, and this is a beautiful organoid that we can take those crypt stem cells and create them in culture. What we found is that when you put those stem cells together with lymphatics, it basically controlled their usage. We could see that the area of those organoids were smaller and their ability to differentiate was much delayed. So what are the signaling that coming from lymphatic that really control stem cell usage? To do that, we did spatial transcriptomics. So just to guide you through, spatial transcriptomics allow us to take spots of those cells, and we basically projected how many or which types of stem cells existed in each point. What we were able to do is to project that now on a 2-dimension axis, and as you can see here, those are the lymphatic vessels in purple. So we could identify the base lymphatics and those that are in the lacteal. So now we can actually ask which factors are expressed on lymphatics that are associated with the stem cells versus those that are not associated with the stem cells. And what we were able to find is that those lymphatic-based, the lymphatics that are the base of the stem cells, highly express known stem cell factors like R-responding 3, but we were also able to identify a completely new stem cell factor called railin. And using genetic models and also functional assays, we found that railin that's secreted from lymphatics is actually important for organoid formation and for stem cell usage within the intestine. And so it turns out that no matter what the usage of those stem cells, lymphatic niche holds stem cells and controls stem cells across epithelial tissues. And this is very exciting time, because while we found the lymphatics are important niche component for the hair follicle stem cells, we also showed that it's across epithelial tissues in the intestine. Other works, although not stem cell centric, have shown that lymphatics are important for brain function, heart development, and very recently showing that lymphatics actually also exist in the bones where hematopoietic stem cells reside, and that is completely new to our understanding. One of the things that we do know is that when lymphatics are dysfunction, or we have excessive or insufficient lymphatic function in a way, that is associated with a lot of regenerative pathologies, and that include lymphedema, chronic wound diseases. We know that during aging, our lymphatics function less and less good. And at the end of the road, cancer, or most of our cancer, originate from those stem cells, and we know that lymphatics are the main exit route for creating metastatic disease. So all of this together, we really understand that there is something to do with lymphatic stem cells that may be targeted to prevent those really hard pathologies that affect the human health. And the problem is that we all know that regeneration is at the end of the road, an arrow with a one negative direction. It will happen to each and every one of us, if not already. We will regenerate our tissues less and less efficient as we grow older. I can see a scratch on my baby, and I see that within a few days almost you can't see anything, but a scratch that I got from my cat is already there for a month, and I'm like, this is not a good sign. And so at the end of the road, what we are spending a lot of time thinking in the lab is it possible to slow aging? Taking those stem cells and tell them to do what they're supposed to do for a longer time point. But maybe to ask a little bit more provocatively, can we reverse it? Can we take those old stem cells and trick them to think that they're young? So how do we know that we're aging? I just mentioned the scratch that I have on my skin, but the skin is really one of the first things that we notice, that we see, that give us the idea that, okay, something is happening in my ability to regenerate, the texture of the skin, the ability to regenerate, and obviously we see some kind of, the hair color is changing. So this is really one of the first things that we notice, oh, I am aging. And why is that important? Because at the end of the road, inability of those stem cells to regenerate will cause chronic wound diseases. And chronic wound diseases, just to give you a little bit of numbers, affect about 6.5 million patients in the United States, and they're considered to be the silent pandemic because we don't really hear about it a lot. And as we age and as we go older and older, that affects more and more older population, and it also predisposed to cancer formation and squamous cell carcinoma that I will mention towards the end. So it's a major health burden that affect so many people in the United States and worldwide. So why are we aging? Are we actually losing the stem cells, or we're losing the environment where those stem cells reside in? So if you have a seed but you don't have the soil, then it will just not grow. So we basically ask the same question in the hair follicle. Why are those hair follicles aging? What happened to them? So we profile them using single-cell RNA sequencing, and the only thing that I want you to just notice is that the picture from the age and the young looks pretty much the same. The same cell population, the same environment, although a little bit less of them, but they're still there. They express exactly the same factors that they used to express in aging, so all the stem cell factors that are important for them are still there. So that was kind of bummer, because we thought that, okay, now maybe we can target them, but they look completely the same. They're there. Why? Clearly, they don't do what they're supposed to do. You can see that the old mice don't grow hair, while the young mice can grow hair. So why do we age? We decided to take the same three-dimensional strategy to look at those stem cells, and you can see that in the young stem cells, those green cells are in a structure that creating kind of beautiful bulge. But when we looked at the old, they're still there, expressing exactly the same marker that the young stem cells expressing, but the structure basically collapsed. They still have their niches, their stroma niches basically on the bottom. They have all the components that they need, but they're just collapsing and not doing what they're supposed to do. So Katya and Madison in my lab were very interested in understanding what happened in aging, and they wanted to understand how lymphatic vessels look like. You can clearly see that the structure of lymphatic vessels as we grow old is completely destroyed. They are much dilated. They are presenting structure that is very, very destroyed. And functionally, they are not able to drain fluids and macromolecules as well as you would expect. But what about the association with stem cells? So the structure is a little bit aberrant, but what happened with the association? What we found is that the stem cells themselves are still there. They're tightly associated still with blood vessels in red, but their association with lymphatic is completely disrupted. They are no longer associated with this important niche that can control their usage. So using the same angiopoietin-like 7 that is expressed by the stem cells to control lymphatic behavior, what we've done was basically to knock down angiopoietin-like 7, and what we found was a mimicking phenomenon, mimicking the lymphatic structure that happened in aging. Their structure were no longer intact. They were very much aberrant in their structure. And what we noticed is, although these two mice were basically litter-mate, they are brothers. One of them was having no angiopoietin-like 7, dysfunctional lymphatics, and they started to lose hair prematurely. So we can push aging fast forward, but can we actually reverse it? So we did a very fun experiment where we took young mice and old mice. We separated the stem cells from each and every one of them, but also their environment, all the cells that surround those stem cells. And we mixed and matched. We put the young stem cells in an old environment and the old stem cells in a young environment, and then we transplant them to the mice and see what happens. What was very weird to see is that the young stem cells in an old environment just did not grow hair. They did nothing. They were still there, but did nothing. When we transplant the old stem cells in a young environment, all of a sudden those old stem cells that didn't grow hair before that were able to grow hair in the recipient mice. So we can trick those stem cells to become young, and now we are trying to really understand if we can trick it in a healthy way. Because one of the problems that we were facing is that when we knocked down the angiopoietin-like 7 that caused the premature aging, we also found hyperplasia of the tissue. So those are very, very big follicles together with small follicles that exist in the skin. So when lymphatics are dysfunctional, stem cells don't know to do what they're supposed to do, but we can also see hyperplastic hair follicle. And that was associated with increase of DNA damage within the stem cells, which suggests to us that maybe stem cells can control lymphatic behavior in order to preserve their genomic integrity and to prevent cancer. And this is maybe why we see more and more cancer predisposition in conditions of lymphatic dysfunction. And that can progress and become squamous cell carcinoma. And again, a little bit of numbers, but it affects so many people in the United States to the point that we have almost 200 people being diagnosed with squamous cell carcinoma every hour, which is a crazy amount of people. Although this disease rarely metastasizing, when it does, it presents a very, very aggressive disease. And cancer doesn't work in a void. We know that the normal cancer adapt the normal environment, the stroma cells, the immune cells, including the vascular system surrounding it, to grow and thrive. And what we found is that the stem cells, those cancer stem cells here in red, were tightly associated with lymphatic capillaries in the squamous cell carcinoma model. And we're trying to understand right now why the cancer stem cells also adapt what normal cells are doing, so we can potentially prevent their ability to see the metastatic disease later on. And with that, my lab really focusing on understanding how epithelial stem cells communicate with their microenvironment to enhance regeneration and prevent diseases. We're using a shield of tools, as you can see here, and we are recruiting if you know anyone that is interested. And just to thank my lab member, I mainly showed results today from medicine, and Katya. I am in the Sanford Consortium, where also Evan's here, and it's an amazing atmosphere to work on regenerative medicine, and I'm very thankful for that, and happy to take questions.